Exercise 2 encompasses wet mount techniques. This is a fun lab where you're going to get to see a lot of those critters that are in the pond water or lake water with you while you are swimming. It's often quite eye-opening, and it's a great lab just to spend some time and take a look at the varied life that's present in water that is not visible to the naked eye. A temporary wet mount is defined as a wet mount made by suspending a specimen of fluid or using a fluid containing a specimen, covering it with a cover slip, and observing it with a microscope. Wet mounts are ideal for viewing living microorganisms. In this lab, you will view a prepared mixture of yeast. The yeast is Saccharomyces. It's plain old bread yeast from a packet, and bacteria, a very common organism known as Escherichia coli, or you may be using Proteus vulgaris. Proteus vulgaris is the most common cause of urinary tract infections for patients that are hospitalized, whereas Escherichia coli is the most common cause of urinary tract infections community acquired, or those individuals who are not hospitalized. There are three objectives while we are working in this lab. First is to know how to use the proper light levels when viewing a wet mount. Second is to compare a eukaryote with a prokaryote. And third is to observe motility and means of motility. Light adjustment is very important when viewing wet mounts. Most living organisms we view in this lab will be colorless, so it's very difficult to distinguish them from their background. In other words, the refractive index of the organism is not that different from the refractive index of water. So we have to make some adjustments to our microscope to ensure that we can see these organisms. When viewing a wet mount, be sure to decrease the light passing through the specimen by adjusting the iris diaphragm. You're going to close the iris diaphragm, sometimes almost all the way closed, and you also will lower the condenser just slightly, about a quarter of the way down. This is going to increase refraction of the light as it comes to the specimen. So the light is bending more when it comes into the specimen, and once it hits the specimen, it will bend even more, allowing you to see sort of a shadow and allowing you to see the specimen in more detail. There are a number of clinical uses of wet mounts as diagnostic tools. For example, an OB/GYN may want to diagnose a vaginal yeast infection, and by taking some vaginal fluid, we'll see these yeast cells. Likewise, an OB/GYN who would want to diagnose bacterial vaginosis will take some vaginal fluid and take a look for these things called clue cells. Clue cells are epithelial cells that are covered with tiny, tiny bacilli. We'll talk about this in Module 5 in lecture, but these tiny bacilli are representative of the organisms that cause BV, or bacterial vaginosis. The syphilis spirochete, Treponema pallidum, is visible with a dark field wet mount. Dark field microscopy is a type of microscopy discussed in Exercise 1 in the lab manual. Also, urine sediment. We'll be doing urine sediment at the end of this semester, and there are quite a few organisms and cells that can be seen in urine sediment. Here's Trichomonas. Trichomonas is a parasite. It's a protozoan that can be seen in urine in a person who has trichomoniasis. Finally, a podiatrist may be interested in a nail scraping, for example, a toenail and a fingernail, to diagnose a nail infection. A podiatrist would use a microscope and something called potassium hydroxide, or KOH. Potassium hydroxide digests away the tissue and it leaves the fungi behind, and we'll see something similar to this image when looking through the microscope. So clinical specimens often are viewed with a microscope by the clinician, even in the office. Now, there are a number of advantages and disadvantages to using wet mounts. So let's look at the good first, the advantages. You can see the natural size and shape of organisms. When we dry and stain organisms, this does change their shape minimally, but it changes their natural coloration due to staining. We can view motility. If the organism is moving about, we can see it moving. And if the organism is large enough and its means of motility are large enough, we can figure out what it is using to move. Is it using cilia or flagella? We can view interactions between organisms. You'll get to see rotifers in this lab, and rotifers are going to interact greatly with their environment. They have a spinning crown of cilia that they move in order to draw organisms to them that they can consume using a vortex. We can assess their means of nutrition. A big clue is colors. If we see green or gold or brown, that tells us the organism has photosynthetic pigments and is getting its nutrition by photosynthesis. And they're quick and easy to prepare. Disadvantages? Well, motile organisms, while it's nice to see it, they can quickly move out of view, so you may be chasing an organism, moving the microscope stage very rapidly, and you might lose it. The specimens are temporary, so they do dry out quickly. We have limited magnification. Oil immersion cannot be used because of the cover slip. Unless you're a very experienced microscopist, you will not be using oil to view these specimens. And of course, live organisms can be hazardous to the user. Referring to that previous slide, a yeast infection or a trichomonas infection. We don't want to contaminate the user, and this is possible when you have a living specimen on a microscope stage. And any type of fluid on the microscope, if you get it onto the lenses, can damage the microscope. In this lab, you'll be viewing motility, and motility is different between prokaryotes and eukaryotes. Prokaryotes are motile via flagella, endoflagella, gas vacuoles, or fibrils. Prokaryotic cells are very small. You will not be able to see these flagella. We will be using a special stain in upcoming labs in order to view bacterial flagella. But in a wet mount, we can infer that they have flagella because they are moving, but we are unable to see them. Prokaryotic motility, as I said, must be inferred. Don't confuse motility with Brownian motion. Brownian motion is caused by water molecules. Water molecules are always moving. They bump into everything. If you're sitting in the bathtub, they're bumping into you, but you can't feel it, and they don't move you about because you're too large. But bacterial cells are very small, and because water molecules are bumping into them, they actually will displace the bacterial cells. So you'll see sort of a shimmering type of an appearance, a motion in there, and that's Brownian motion. Brownian motion is not a means of motility. Now, eukaryotes have a variety of means to move about. Some of them are non-motile, but some have cilia. Cilia may be fused together. We call these cirri. Flagella. Flagella are used to move eukaryotic cells. They are much thicker in diameter. They can be seen with a light microscope at 400x magnification. Pseudopods, or false feet, are found in amoeba. They're going to extend these pseudopods. You'll be able to see amoeba moving in this lab. Some eukaryotes move by contraction of muscles, for example. Others move by appendages. And finally, a eukaryote, known as a diatom, moves by secretions from a raphe. A raphe is a slit that is in a siliceous test or shell of the organism, and crystalline material is going to ooze out of that raphe, and the organism is going to glide over the surface. So what to expect in this lab? First, in the first period, you're going to look at a mixture of yeast and bacteria. Yeast are going to be spherical or ovoid in shape. They are larger than bacteria. Looking in between the yeast cells, you're going to see these tiny dashes. These are the bacteria. Take a look to see if they are motile. Look for Brownian motion. Are the yeast affected by Brownian motion? Are they motile? Are the bacteria motile? Are they affected by Brownian motion? As you are looking at these specimens, try to determine this. In both period 1 and period 2, you're going to be looking at pond water. I will have many samples for you to look at. You're going to be doing something called a hanging drop wet mount. I'll show you how to do that in lab. We're also going to have special samples in these slide chambers that are seen on the bottom. I'll also demonstrate how to use those. They're a lot of fun to look at. A lot of different organisms will be in there. You are going to try to identify at least 6 different organisms during the lab. Draw pictures of them. Try to identify them with the dichotomous keys that you're given in lab, as well as the lab manual images and other images that I provide in lab. Try to identify them before you leave lab. Once you draw the picture and you go home and try to figure out what it is, you're going to have some difficulty. Looking forward to seeing you in lab.